మోస్ట్ ప్రాబ్లీ నేను స్కూలింగ్ చేసింది ఇక్కడనే మేడం తూప్రాన్ అని ఇక్కడ నుంచి ఒక 20 కిలోమీటర్స్ ఉంటుంది. అక్కడైనా రావచ్చు, లేదంటే లేదు పిఎస్సిస్ ఆర్ మన గాంధీ ఉస్మానియా నీలోఫర్ కోటి మెటర్నిటీ ఉన్నాయి కదా, అక్కడ రావచ్చు. లేకపోతే కమ్యూనిటీ హెల్త్ సెంటర్ లేకపోతే ఏరియా హాస్పిటల్స్ ఎక్కడైనా ఇవ్వచ్చు.
Streaming start.
Yeah, started. Yeah. కొద్దిగా టైం పడుతుంది. సిస్టమ్ స్లో అయిపోయింది. వన్స్ ఇట్ గెట్స్ మొమెంటం స్పీడప్ అయిపోతుంది. బస్లో రారా మీరు?
నేను కార్లో వస్తాను సార్.
ఎక్కడ ఉంటారా?
ఇక్కడ గుండెపోచంపల్లి సార్, కొంపల్లి.
గుండెపోచంపల్లి.
Okay, good morning students. In continuation of the lipid metabolism, small few topics are left over that I would like to finish off today. So now let us discuss about the prostaglandins. Okay, if you remember your previous classes of lipid metabolism, so already we have done with the chemistry of lipids, so how they have been classified. Okay, and then we have already finished the digestion and absorption of the lipids, how the dietary lipids, how they'll be digested and absorbed. And even we have seen the like structure of the cholesterol and what are the derivatives of cholesterol. Okay, and already you know the derivatives of cholesterol are all steroid hormones, vitamin D and bile salts and bile acids. So these are the derivatives. These are the important biological substances which are derived from the cholesterol, and you know that cholesterol is a derived lipid. Okay, and it's a 27 carbon compound, and the structure is described as cyclopentanoperhydrophenanthrin ring. Okay, and if you see the structure, there will be a hydroxyl group at the 3rd carbon. Okay, and double bond between the 5th and 6th carbon. There is a side chain at the C17. Okay, so like that you have to describe the structure of the cholesterol and mention its derivatives like steroid hormones, all steroid hormones, and the vitamin D, which has a major role, which plays an important role in calcium and phosphorus metabolism, and even the bile salts and bile acids, which plays an important role in digestion and absorption. So all these biological important compounds are derived from cholesterol. Now coming to the next one is prostaglandins. Okay, prostaglandins are also examples of derived lipids, and these all the compounds which are like prostaglandins, they are collectively known as eicosanoids. Okay, prostaglandins and related compounds, okay, all these are collectively known as eicosanoids, and these are produced, okay, these prostaglandins are produced from arachidonic acid. Okay, you know that essential fatty acids. Okay, so in the chemistry we have discussed that essential fatty acids means these are essential, but they cannot, why they are essential means they cannot synthesize in your body, so they need to supply through diet. So those fatty acids are called essential fatty acids. So examples of essential fatty acids are arachidonic acid, linoleic acid and linolenic acid. These three are called essential fatty acids right here. So out of that, the one of the essential fatty acid, that is arachidonic acid. Okay, arachidonic acid is how many carbons are there? It's a polyunsaturated fatty acid. Okay, I think you know the meaning of polyunsaturated fatty acid. In their structure, which is having more than one double bond, that those type of fatty acids we can call it as polyunsaturated fatty acids right here. One of the example of a polyunsaturated fatty acid is arachidonic acid. Okay, this is the precursor. That is, precursor means that is the starting material for the synthesis of the prostaglandins right here. Eicosanoids means, okay, this is the term eicosanoids means these are the prostaglandins and their related compounds together known as eicosanoids, and these prostaglandins or eicosanoids are derived from like a polyunsaturated fatty acid, that is essential fatty acid, which is arachidonic acid. Okay, and the eicosanoids, they are considered as local hormones. Okay, that means their mechanism of action is like they may be act like hormones, because these eicosanoids, they have like a specific target cells, okay, close to their site of the formation where they are formed. There only they will act upon the specific target cells. Okay, even hormones also, they will be synthesized in one place, and their mechanism, they will exhibit their action on other site on the target cells. Like that, even prostaglandins also, they will act like hormones because they will be produced all in one place. There only in nearby that only their target cells will be there. There only it will be acting, and these prostaglandins are rapidly catabolized, means they are rapidly degraded, so they are not transported to distal sites within the body. Okay, so that means here wherever they are synthesized, immediately in few minutes only they may be degraded. So that's what, they cannot be transported to, or as such they cannot be transported to all sites in the body. Okay, examples of the eicosanoids are, see here, examples what we have learned here, prostaglandins and related compounds are called eicosanoids. They are derived from arachidonic acid. Okay, and they may act like local hormones because wherever they are synthesized, there only on the specific target cells they will be acting, and they are rapidly degraded. That's what prostaglandins, their action is like restricted to that place only. Okay, and examples of these eicosanoids are, please note down, prostaglandins, prostacyclins. Examples of eicosanoids are prostaglandins, prostacyclins, thromboxanes, leukotrienes, and epoxycosterinoic acids. Okay, you just remember these four names. Examples of eicosanoids means, what are eicosanoids? I mentioned prostaglandins and their related compounds are known as eicosanoids, and eicosanoids examples are prostaglandins, prostacyclins, thromboxanes, okay, leukotrienes. Okay, these are all the examples of eicosanoids. Then concentrate here because prostaglandins is important. 4 marks question for you. So whatever I am showing here in the slides, you have to write it in the paper when it is asked for 4 marks. Okay, and what are the functions of the eicosanoids means, mainly they plays an important role in inflammation, fevers, and the regulation of the blood pressure, and even they have a role in blood clotting process and immune system modulator. These eicosanoids acts as immune system modulators, and they can control of the reproductive process and tissue growth. Okay, and even the regulation of the sleep and wake up cycles. Okay, these eicosanoids has the diurnal variation, means they have a role in the regulation of the sleep and wake up cycle. They may regulate your blood pressure, has a role in the blood clotting and immunity, and it has a role in the reproductive process and tissue growth, and even this eicosanoids has a role in inflammation and fevers. Okay, these are the functions of the like eicosanoids, and the prostaglandins, prostacyclins, thromboxanes and leukotrienes are the examples of eicosanoids. Okay, now here these are the prostaglandins. I have already mentioned these are all most biologically active substances. Okay, and they, you know that they are all considered as local hormones because of the target cells they may act. Okay, and you know that these prostaglandins are derived from a polyunsaturated fatty acid, that is arachidonic acid. And different types of prostaglandins are already, you know that like prostaglandins are, there are different types are there. PG is prostaglandin. See just here, see the eicosanoids. One of the eicosanoids examples are prostaglandins, prostacyclins, thromboxanes and leukotrienes. That means prostaglandins comes under eicosanoids. Even under that prostaglandin only, there will be different types of the example. Prostaglandins are of different types are there, that is prostaglandin A, B, okay, that is PG represents prostaglandin A, prostaglandin B, E and F. And here these prostaglandins belongs to different series. Okay, like the series 1 means which might be having only one double bond, 2 means which may be having 2 double bonds. Usually the common ones are PG2, that means 2 double bonds are there in their structure, and few series are there like 3 double bonds are there, those will comes under series 3. The widely distributed prostaglandins are PGD2, E2, I2 and thromboxane 2. Okay, see here these are the different types of prostaglandins. Okay, different types of prostaglandins are already mentioned, A, B, E and F. Okay, and there are different series are there, 1, 2, 3 based on their double bonds. If one double bond is there, those will comes under series 1. If 2 double bonds are there, 2 like they will comes under series 2. And if 3 are there, they will comes under series 3. Most important ones, existing ones are like prostaglandin 2, that is 2 double bonds are there. The widely distributed prostaglandins are, please note down, PG, that is prostaglandin D2, A2, I2 and thromboxane 2. Okay, and this is the structure of the PGE2. Okay, this prostaglandins all have this like this. If you see the structure, all the prostaglandins, their structure may be like this. Cyclopentane rings are there. Okay, and what is this E? E, D, E that mentioned, no? So the letter code that is based on the ring modification. Okay, based on the ring modification, the prostaglandins have been given the letter code, that is A, B, D, E, etc. And there is a subscript 2 is there. Already you know that represents the number of the double bonds in the side chain. Okay, so see here there are different types I mentioned, no? That is PG, D2, E2, I2, okay, D, E, E. What are those letters? They may be represent to the ring modification. This is the general basic structure, cyclopentane structure of the prostaglandin. Based on the ring modification, there is like a letter code may be there, D, E, I, etc. And there is a subscript is there, 2, 1, 2. Okay, if it is 2 is the common one, that 2 represents the number of the double bonds in the 2 side chains. And one of the other type of eicosanoids is thromboxanes, and these are similar, but instead of the like 5-membered ring, in thromboxanes there is a 6-membered ring is there. Okay, now coming to how the prostaglandins are derived. So first I mentioned all the different types of eicosanoids are derived from the precursor, that is arachidonic acid, which is also known as arachidonate. Okay, and this arachidonate comes from diacylglycerol. What is this diacylglycerol? Fatty acids, triglyceride fats, that is triglycerides, which hydrolyze to DAG, that is diacylglycerol. This may be converted to arachidonic acid, and even the phospholipids may be converted to arachidonic acid. So that means arachidonic acid is the precursor for the formation of eicosanoids, and this arachidonic acid is in turn derived from diacylglycerol or from phospholipids. Now here this arachidonic acid may undergo linear pathway catalyzed by the enzyme lipoxygenase. It may be converted to one of the eicosanoid, that is leukotriene. Okay, or sometimes this arachidonic acid may undergo cyclic pathway catalyzed by the enzyme synthase to form prostaglandin H2. Okay, and this prostaglandin H2 by the enzyme prostacyclin synthase, they may be converted to prostacyclins, or prostaglandin H2 by the enzyme thromboxane synthase, they may be converted to thromboxanes, or sometimes prostaglandin H2 may be converted to other prostaglandins. Okay, I think you are following this. So that means icosanoids, prostaglandins and their related compounds are known as icosanoids. What are the examples of the icosanoids? Examples are leukotriene, prostaglandin, prostacyclin and thromboxanes. 1, 2, 3, 4. These 4 are the examples of icosanoids. So these icosanoids are derived from which compound? Arachidonic acid. Okay, this arachidonic acid is derived from fat, that is diacyl glycerol, or from the phospholipids. This arachidonic acid has been formed, and this arachidonic acid may be undergo 3 different types of the pathways by the enzyme lipoxygenase. By linear pathway, it may be converted to leukotrienes, or it may be catalyzed by the enzyme PGH2 synthase by the enzyme cyclic pathway. By the cyclic pathway, this may be converted to prostaglandin H2, right? And prostaglandins by the enzyme thromboxane synthase may be converted to thromboxanes, or prostaglandin H2 catalyzed by the enzyme prostacyclin synthase to form prostacyclins, or prostaglandin H2 may be converted to other prostaglandins. Like that, from arachidonic acid, that is polyunsaturated fatty acid, all the prostaglandins and the different types of icosanoids have been derived. Already we have discussed the functions. They're like the functions of these icosanoids or prostaglandins, that they have the function in the blood pressure. They regulates the blood pressure, and they maybe regulates the immune system, control the reproduction, fever, inflammation, sleep wake up process. Like that they are synthesized. Okay, and that's it about the, then here coming to the functions of the prostaglandins. Again, please note down the individual functions is prostaglandin I, that is also known as prostacyclins. Prostacyclins are known as prostaglandin I, which is a vasodilator. That's what it is used in regulating the blood pressure, and it inhibits the aggregation of the platelets. And thromboxanes, that is a vasoconstrictor, activates the platelet aggregation. See here, thromboxane function is just quite opposite to the prostacyclins. Okay, prostacyclins are vasodilators, inhibits the platelet aggregation. And thromboxanes are vasoconstrictors, they may activate the platelet aggregation. And prostaglandin F2, that stimulates the uterine muscles, and it is the bronchoconstrictor. Okay, see, I already mentioned what is this XIFE, that is the, these are the based on the ring structure. Okay, prostaglandin has been mentioned with the code, that is IFX. Okay, XFED, that codes represents the structure of the ring. Okay, and this subscript 2 represents the number of the double bonds in its structure. But each prostaglandin has its own function. PGI has a vasodilator, inhibits platelet aggregation, and PGF2, that stimulates the uterine muscles, and it is a bronchoconstrictor. And PGE2, that is a bronchodilator. PGE2 is a bronchodilator. Okay, and PGD2, it produces inflammation. And thromboxanes, it is a thromboxane, that is TX2. It is a vasoconstrictor, and it activates the platelet aggregation. Okay, these are the functions of the prostaglandin. Please note down this function. This question, functions of the prostaglandin, is important 4 marks question. Okay, and here if you want details, PGE2. Okay, other functions is PGE2. Here PGE2, it relaxes the smooth muscles in blood vessel and respiratory system, and it constricts the smooth muscle in female reproductive tract, and it constricts the longitudinal muscles in the GI tract, and it increases the renal filtration. And okay, these are the functions of PGE2. PGI2, already I mentioned, PGI2 is nothing but a prostacyclin. It's a vasodilator, relaxes smooth muscles, especially of the arteries, uterus, and it constricts the smooth muscles of the GIT tract, and it inhibits the platelet aggregation, and it increases the renal filtration. And okay, other functions of PGF2 as it constricts a smooth muscle in every system, and it is a bronchoconstrictor. Okay, then coming to the thromboxane A2. Okay, thromboxane A2 here it is, it's a bronchoconstrictor and it enhances the platelet aggregation. Okay, and these are the main functions of the like prostaglandins. And coming to the other one is leukotrienes. It is one of the icosanoids. It is produced from arachidonic acid. It is a potent chemotactic agent. Okay, and it slow down the rate of the anaphylaxis. Okay, slow reacting substance of anaphylaxis. And here leukotrienes may cause smooth muscle contraction, bronchoconstriction and increased capillary permeability. Okay, leukotrienes function is they may cause smooth muscle contraction and bronchoconstriction, and they may increase the capillary permeability. Okay, that's it about this prostaglandins and related compounds, that is like icosanoids. Okay, here in this you have to concentrate on the like what are the prostaglandins and their functions. Okay, and then the different types of the icosanoids. Okay, then we'll move on to the next topic. Give me 1 minute time, I'll change the slides. Okay, next topic I am going to explain you is atherosclerosis. Okay, atherosclerosis means, first before explaining this atherosclerosis, first you should know about the heart. You know, it's a muscle of the size of your fist and it weighs about 1 pound, and it is located and slightly left to the like breastbone, and yearly it pumps about 4.7 liters of the blood every minute. Okay, and here this is the anatomy of the heart. And the function of the heart is to you know that it is circulate the blood throughout the body by pumping. Mainly function of the like heart is to circulate the blood by pumping the blood through the lungs. It removes carbon dioxide and it refreshes the blood with the oxygen, and the oxygenated blood is pumped to the body to provide oxygen and nutrients and remove the waste products. And the coronary arteries are the main blood vessels that supply blood and oxygen to the heart muscles. Okay, now here if you see the blood supply to the heart, there is two coronary arteries are there here. They will be branched from the main aorta that is present just above the aortic wall, and here this this aortic valve is like larger than the drinking straws. Okay, and they may be divided into and they may be encircle, okay, the heart and to cover its surface, and they may form a network like substance. Okay, that is reminded the physician of a slightly crooked crown. Okay, that means coronary comes from the Latin word coronarius. Okay, see here this there is these arteries, they will be like encircled the heart. Okay, and they will they carry about the 130 gallons of the blood through the heart. Okay, and this is the blood supply to the heart. Now here coronary artery disease, CAD, is the one of the most common and serious effects of the aging, and fats, they may be deposited in the blood vessel walls. Okay, here what happened in the coronary artery disease, excess fat is there that may be deposited in the walls of the arteries, coronary arteries, and they prevents the free flow of the blood. Okay, so the blood which is carrying oxygen, okay, that is not supplied enough to the muscle, especially to the heart muscle, that resulting in the atherosclerosis that often can leads to eventual blockage of the coronary artery and leads to heart attacks. Okay, see here in the coronary artery disease what happened, the fatty acids are deposited in the blood vessel walls, and they may be narrow the passage of the movement of the blood. I mean, as it is a tube like one, as it is deposits, it prevents the passage of the blood. So the resulting condition is called atherosclerosis. Okay, and often that may leads to heart attacks. Now here this is the, see the picture, this is the diseased artery. Now here mortality is CAD, cardiovascular diseases are claimed to the 39.5% of all deaths, or 1 of every 2.5 deaths in United States in 2000 or because of CAD, cardiovascular diseases. And here cardiovascular diseases was this is the major cause for the deaths. And here see this picture here, these are the coronary arteries. See here, this is the artery, atherosclerosis with the blood clot, okay, and this is the atherosclerosis, okay. That means what is atherosclerosis? Nothing but the deposition of the fat, deposition of the various types of lipids in the blood vessels prevent the free flow of the blood, okay. So this is atherosclerosis. On the top of it blood clot is there, and here the atherosclerosis, and this is the spasm. That all these conditions prevent the free flow of the blood. And here now here see next one is here the signs and symptoms of this CAD are, so this is the referred as a silent ischemia, okay, and blood to your heart may be restricted due to coronary artery disease, but you don't feel any effects, okay. See here atherosclerosis means here fat may be deposited in the coronary arteries, that may cause ischemia. Enough blood is not pumped, okay, not supplied to the heart muscle, so you may not feel like any effects. Chest pain is there if any coronary arteries cannot supply enough oxygen, enough blood to meet the oxygen demands of the heart, that may be results in the chest pain called angina, okay. First here initially you don't feel any effects if there is CAD, and here sometimes here sufficient blood, ischemia is there, sufficient blood, oxygen is not supplied to the heart, that may result in the chest pain called angina. And there is shortness of the breath is there. Some people may not be aware that they may be having coronary artery disease because they may develop the symptoms of congestive cardiac failure, okay. That means extreme fatigue with the exertion, shortness of the breath, and swelling in their feet and the ankles. So heart attacks results when the artery that supply blood to the heart muscle becomes completely blocked, and the party of the, and here the heart muscle may be lack of oxygen, that may be dies, that may be leads to coronary artery diseases, okay. Now the signs and symptoms are none initially: chest pain, angina, shortness of the breath, heart attack, etc. Now here atherosclerosis means here atherosclerosis can and does occur in almost any artery in the body, okay. Means it may like fat may be deposited in any artery in the body, but in the heart its effect can be crucial, okay. The body depends on a strong pumping of the heart to circulate the life-giving blood, and this includes the heart muscle itself, okay. See here the fat may be deposited in any artery, but the impact will be there if it is deposited in the coronary arteries, because coronary arteries are the main ones which supply blood to the heart, okay. And if that one will be affected, whole the pumping, okay, the main function of the heart is to pump the blood, that may be affected, okay. And here in the coronary artery, if the coronary arteries become blocked, the cardiac muscle begins to fail, and so the blood circulation decreases, which includes the circulation of the heart muscle itself, okay. Now here the blockage, here this is a picture which is showing the blockage in the right coronary artery, okay. So this is the blockage in the right coronary artery, okay, because of the deposition of the fat or cholesterol. And the causes for atherosclerosis may be high blood cholesterol, high blood pressure, smoking, obesity, and lack of physical activity. These are the main causes for this, okay. If there is excess cholesterol is there, that may be deposited, that may be forms flakes deposited on the walls of the arteries and forms flakes, causes ischemia. If high blood pressure is there, okay, smoking, obesity, and lack of physical activity, that may be the causes for the like atherosclerosis, okay. And the risk factors are, because few are controllable and few are you cannot control. Like if it is risk factors may be because sex, hereditary, and race, age. So these are the uncontrollable factors, but the controllable factors are you can like high blood pressure, high blood cholesterol, smoking, physical activity, obesity, diabetes, stress, and anger. If you all control this one, you can decrease the risk of the atherosclerosis. Now here, how do you screen and diagnosis means? The first one is initially electrocardiogram, ECG. So that will measure, okay, that will measure the electrical impulses of the heart. And there is a stress test is there that measures the blood supply to the heart. And there is coronary angiography is there that shows the specific sites of the narrowing of the coronaries. Okay, see here. Like that you can screen initially by the ECG. What ECG will show? That means electrical impulses. It will measure Okay, and the next stress test is there, that will measure the blood supply to the heart, and coronary angiography is there, and that shows the specific sites of the narrowing of the coronary arteries. Okay, and here the other tests includes our blood test. Generally in the blood test we do evaluate the kidney and thyroid function, as well as to check the cholesterol levels and the presence of anemia. Okay, so generally for atherosclerosis the blood test will order is thyroid functions, kidney functions, and also to check the like cholesterol levels and the presence of anemia. And here the chest x-ray shows the size of your heart and whether it is a fluid build up around the heart and lungs. And echocardiogram, it shows the graphic outline of your heart movements, and ejection fraction determines the how well your heart pumps, okay, with the beat. These are the other tests including this atherosclerosis. So treatment is, many people are able to manage the CAD with the lifestyle changes and the medications, and other people with severe coronary artery disease may need angioplasty or surgery. Okay, initially if it is diagnosed in the early stages, by changing the lifestyle changes and medication, we can manage this CAD, and maybe if in severe cases need angioplasty or surgery. Okay, this is the angioplasty. See here the blocked coronary artery, so there may be by it can be treated by applying the stent. Okay, this will disperse the fat which is deposited in the coronary artery, and the balloon blasting. Okay, so this balloon will be inserted. Once it go to the heart, it will be blow, and that will means clear the fat which is deposited in the coronary arteries. Okay, this is the narrowed artery which can be treated by like the stent and balloon therapy. Okay, and the stenting means already you know, a stent is introduced into the blood vessel on a balloon catheter and advanced into the blocked area of the heart artery, and the balloon is then inflated and causes the stent to expand until it fits into the inner wall of the vessel, conforming the contours as needed. Okay, so here there is a stent will be introduced, and once it goes through the artery, there it will be inflated, and that stent will fit into that area, that prevents a further deposition of the like fat and prevents a ischemia. And the balloon is then deflated and drawn back. The stent stays in a place permanently, holding the vessel opening and improving the blood flow. Okay, the stent will be wherever we introduce, so there only it will be permanently stay and prevents a further clotting of the blood. Okay, next one is angioplasty, other treatment for atherosclerosis. Here a balloon catheter is passed through the guiding catheter to the area near the narrowing, and here a guide wire is inside. The balloon catheter is advanced through the artery until tip beyond the narrowing. So the angioplastic catheter is moved over the guide wire until the balloon is within the narrowing segment. Okay, and the balloon is inflated, compressing the plaques against the arterial walls. Okay, so the place we will like insert the balloon. Once the plate is reached, it will be inflated, and once that inflated, the fat which is deposited over that is compressing the plaques, so that like that it improves the blood flow. Okay, like that. And the next one is the uncontrolled one. If it is not treated, managed by these two, we have to go for the bypass surgery. Here healthy blood vessel is removed from the lung, arm or chest. So blood vessel is used to create new blood flow path in your heart. Okay, so the bypass graft enables blood to reach your heart by flowing around the bypassing. Okay, why it is called bypass means here the blood to reach your heart by flowing around, bypassing the blocked portion of the diseased artery. Okay, so that is we are bypassing the flow. So the increased blood flow reduces the angina and the risk of the heart attack. Okay, that's it about this one. So these are the treatment of this atherosclerosis. So prevention is better than cure here. So get regular medical checkups and control your blood pressure, check your cholesterol regularly, and don't smoke, exercise regularly, maintain a healthy weight, and eat a heart healthy diet, and manage the stress. Okay, these are the prevention for the atherosclerosis. Okay, regular exercise is there. Stress is also one of the major cost for this one, and even the regular monitoring of your blood pressure and cholesterol also one of the cause which prevents heart attacks. Okay, that's it about this one, atherosclerosis. Then we will okay, and the next one is here there is a lipoproteins are there. Okay, next one of the topic in your lipid metabolism is lipoproteins. I'll show you the lipoproteins. Right, give you 1 minute time. Yeah, then coming to the another topic of this lipid metabolism, that is lipoproteins. So lipoproteins are like complex lipids. Complex lipids means along with the lipids, these are the lipoproteins or the complex proteins. Okay, that means proteins in combination with the non-protein part. And even the lipoproteins are also comes under complex lipids. So here lipids along with the proteins to form lipoproteins, because you know that here lipids are insoluble in water. As they are insoluble in water, they cannot transport alone in the blood circulation. So always they will be combining with the protein to form lipoproteins. So lipoproteins are the proteins which help in the transport of the lipids in your blood. Okay, lipids are transported in the blood from one place to another place through in the form of lipoproteins. What do you mean by lipoproteins? Lipids in combination with the proteins is called lipoproteins. These lipoproteins help in the transport of the proteins, transport of the lipids, right, from in your blood circulation. Okay, and these lipoproteins mainly their function is to transport the lipids in the blood, and these are the molecular complexes which is composed of lipids and proteins. And these lipoproteins, based on their composition, okay, based on their density, they have been classified into 5 major classes. Okay, so based on their weight density, they have been classified into kilomicrons, VLDL, LDL, HDL, and free fatty acids with the albumin. Okay, right here there are mainly 4 types of lipoproteins are there. You just remember, lipoproteins means lipids in combination with the protein, that is lipoprotein, and lipoproteins which help in the transport of the lipids. Different types of the lipids according to their density. Okay, if you like kilomicrons, very low density lipoproteins, that is VLDL. LDL, that is low density lipoproteins, HDL, high density lipoproteins, and free fatty acids with the albumin. Okay, right here when you supply, if the serum blood is subjected to electrophoresis, okay, and there are these chylomicrons and ultracentrifugation, these lipoproteins, they will be separated like this. Okay, so first one is the chylomicrons, least density one, then the very VLDL, very low density lipoprotein, intermediate density lipoprotein, LDL, low density lipoprotein, HDL, high density lipoprotein, free fatty acids. Okay, so you just, what is VLDL is very low density lipoprotein, IDL is intermediate density lipoprotein, LDL is low density lipoprotein, HDL is high density lipoprotein. Okay, and even the plasma is subject, even these lipoproteins, different types of lipoproteins can be separated by electrophoresis. Electrophoresis means the movement of the charged particles under electrophoresis. These lipoproteins can be separated into 5 bands. What are the bands which will be obtained on electrophoresis are chylomicrons, LDL, VLDL, HDL. Okay, and so main you just remember important lipoproteins are 4. Based on their density, they will be classified into 4 types: chylomicron, VLDL, very low density lipoproteins, LDL, low density lipoproteins, HDL, and high density lipoproteins. Now here the function of the lipoproteins will be like, the structure will be like this. Okay, see here, and all the protein in the lipoprotein, protein part will be there in the outer surface, and here the lipids are there in the inner surface. Like that, they will be help in the transport of the lipids. Okay, and here then coming to the individual functions of the lipoproteins. The first one is the chylomicron. Chylomicrons is one of the lipoprotein. Please note down this lipoproteins and their functions is also important short answer question. Okay, if the question is asked, you just write lipoproteins means lipids plus proteins. They help in the transport of the lipids. Based on their density, they have been classified into 4 types: chylomicrons, VLDL, LDL, and HDL. Okay, and what are chylomicrons? Chylomicrons are one of the lipoproteins. They may be synthesized in the small intestine. What is the function of the chylomicron? They transport the exogenous, means dietary lipids from the intestine to peripheral tissues. Okay, I repeat, chylomicrons are synthesized in the liver. Their function is to transport dietary triglycerides from small intestine to peripheral tissues. Then coming to the second one is VLDL, that is very low density lipoprotein. It is produced in the liver and intestine. It transports the endogenous triglycerides. Right here, what is triglyceride? You know the structure. 3 molecules of fatty acid combined with the 1 molecule of glycerol to form a triglyceride. Okay, 3 fatty acid with the 1 glycerol to form a triglyceride. So this VLDL, where it is synthesized in the liver, it transport triglycerides from the liver to peripheral tissues. Endogenous triglycerides from the liver and intestine to peripheral tissues. Now what did you understand? Chylomicrons and VLDL, these are the 2 lipoproteins which are concerned with the transport of triglycerides. Okay, fats which are present in your blood, how they will be transported means with the help of 2 lipoproteins, that is chylomicrons and VLDL. Okay, chylomicrons, where they have been synthesized in the intestine, what is their function? They will be transporting the dietary triglycerides, the fat which you have taken through the diet. They will be transported from the intestine through the peripheral tissues with the help of chylomicron. Then the second type of lipoprotein is VLDL. What is its function? VLDL help in the transport of the endogenous triglycerides, not the dietary. The triglycerides which has been synthesized in your blood, in the body, that may be transported from the liver and intestine to the peripheral tissues with the help of VLDL. Then coming to LDL, that is low density lipoproteins which are formed from VLDL in the blood, transport cholesterol from liver to other tissues. Okay, LDL means it transport this VLDL, very low density, this LDL is derived from VLDL. The transport cholesterol Okay, the LDL transport cholesterol from where to where? From liver to other tissues. Okay, but the breakdown, catabolism of the cholesterol, where it happens in the liver. Okay, but here the liver LDL is taking cholesterol from the liver to the peripheral tissues. Once the cholesterol reaches the peripheral tissues, it cannot be undergo degradation. That's what LDL is called bad cholesterol. Why it is called bad cholesterol means here LDL takes it transports the cholesterol from your liver to other tissues. Only cholesterol degradation happens in the liver, but it is not allowing to degrade the cholesterol. It directly transports the cholesterol from the liver to the other tissues. So that's what LDL is called bad cholesterol. And the next one, what is the next one? HDL cholesterol. What is HDL cholesterol? It is mostly synthesized in the liver. HDL, okay, high density lipoprotein. It is synthesized in the liver. Its function is to transport cholesterol. It collects the cholesterol from the peripheral tissues to the liver. Okay, that is reverse cholesterol transport. So once it collects all the cholesterol from your body and transport it to the liver, nicely once the cholesterol reaches the liver, its degradation takes place, then thereby decrease the circulating cholesterol levels. That's what HDL is called, good cholesterol. Okay, please note down. There are 4 lipoproteins are there. Okay, what are they? Chylomicrons, VLDL, LDL, HDL. Chylomicrons and VLDL both help in the transport of triglycerides, fat. Okay, and LDL and HDL, they help in the transport of cholesterol. But HDL transports cholesterol from the peripheral tissues to the liver. So that's what it is called good cholesterol, because the catabolism, the degradation of the cholesterol happens in the liver. And LDL cholesterol, it transports cholesterol, it collects the cholesterol like from the liver to the peripheral tissues. So LDL cholesterol is called bad cholesterol. And even other type of this one is lipoprotein is free fatty acid with albumin, and each molecule of albumin holds 20 to 30 molecules of fatty acid. Okay, so that's it about this lipoproteins. This much details are enough for you, lipoproteins. Now here coming to the next one is here the your lipid profile. So lipid profile means generally we do order lipid profile. Okay, lipid profile means these are the test which will comes under to check the body fat levels. The test we will order we will do in the diagnostic labs is lipid profile. Profile means it's a group of tests which will assess the lipid content. What are the tests which will comes under LF like lipid profile are, please note down: total cholesterol, HDL cholesterol, LDL cholesterol, and then triglycerides. Okay, mainly the tests which will comes under lipid profile are total cholesterol. Okay, I mean the cholesterol is distributed in the HDL and LDL because these two are the transport forms. So LDL plus HDL together known as total cholesterol. So test which will comes under lipid profile are total cholesterol, HDL cholesterol, LDL cholesterol, and then we do estimate the triglycerides. Okay, and now generally the normal cholesterol concentration in the blood should be less than 200 milligram per deciliter. Okay, the reference range, the normal value of the cholesterol should be less than 200 milligram per deciliter. HDL cholesterol is, it should be more than 40, okay, or more than 50 milligrams per deciliter. More the HDL cholesterol, more will be the heart health. Okay, and your LDL cholesterol, okay, low LDL cholesterol should be less than 100 milligram per deciliter. Okay, 80 to 100. And the next one is triglycerides. Triglycerides should be less than 150 milligram per deciliter. These are the normal reference ranges of the lipid profile. What are the tests which will comes under lipid profile? Total cholesterol, HDL, LDL, and then triglycerides. Triglycerides should be less than 100 or 150 milligram per deciliter. Cholesterol should be less than 200 milligram per deciliter, and the LDL cholesterol should be less than 100, and HDL should be more than 40 to 60 milligram per deciliter. These are the tests which will come under the lipid profile. Okay, that's it with the, I think I have finished all the topics which are there in the lipid metabolism. Okay, the lipid metabolism, you may be having the digestion absorption essay question, and classification of the lipids, whole thing is an essay question for you, or the phospholipids, compound lipids, phospholipids is there, 10 functions are there for phospholipids. It is an important essay question for you. Phospholipids, essential fatty acids, that is important short answer question. And the next one is here, lipoproteins and their functions, important short answer question for you. And the next one is atherosclerosis for 4 marks for short answer. Prostaglandins, different types of prostaglandins and their functions, it's a short answer question. And then the structure of the cholesterol and the derivatives, it is also one of the short answer question for you. And the next one is lipid profile and its normal ranges, that is also important, very short answer question. Lipid profile, it's a test with the normal range is the important, very short answer question. Okay, I think with this I have finished all the lipid metabolism. So as far as our knowledge, we have finished all the chapters which is there in your syllabus. Cell, transport mechanisms, then we have finished acid base balance, chemistry, carbohydrate metabolism, lipid metabolism, protein metabolism, enzymes, and then nucleic acid chemistry we have finished, immunoglobulins we have given, and the antioxidants, free radicals, we have given you the notes for that, and even the analysis of the normal urine, and the functional tests kidney function test, liver function test. I think these are all the topics whatever mentioned in your syllabus we have finished as for our knowledge. Okay, if anything we have not finished, you just let us know, we'll finish up that. As per our knowledge, we have finished all the different types of the all the topics which has been there in your syllabus. Okay, take me a 5 minutes break, and I'll continue the just take 2 minutes break. I'll be back. I think here in protein metabolism, I have not finished collagen structure and gelatin. Let me finish up that one. Okay, and connective tissue proteins, this connective tissue proteins are the basic units of the life, okay, and cells which are surrounded by a complex extracellular matrix, often referred to as extracellular matrix, which is also known as connective tissues. Okay, now here in this class I am going to discuss about this connective tissue proteins. Means here the cells which has been covered by a complex matrix like substances which are known as the cells. You know that they are the basic units of the life. They have been surrounded by a complex extracellular matrix which is known as extracellular matrix, also known as connective tissues. This connective tissues has many functions, and they may be mainly help in the supporting of the cells which has been surrounded. And this extracellular matrix may constitute the non-cellular components of the extracellular matrix contains the compounds which are not in the cell and organs, and they plays an important role in the cell differentiation, development and maintenance of the tissue and organ. Okay, you know that extracellular matrix means they are not the components of the cell, or they are not the components of the organ. So these are the matrix like substances which has been surrounded the cells. They may play an important role in the development and cell to cell interaction and differentiation of the cells, and biochemical and mechanical properties of each organ of that depends upon the composition of the extracellular matrix. And post-translational changes of the extracellular proteins, they may be significant in function. Okay, so the main specific function of the organ that depends upon the composition of the extracellular matrix. So the main components of the extracellular matrix are water, proteins and polysaccharides. So what are the components which are there in the extracellular matrix? Means water, proteins and polysaccharides, and their composition. And the composition of each tissue is unique to suit the functions. Okay, and here that's what each cell has been surrounded by different extracellular matrix that will determine the function of that particular cell or function of that particular organ. Usually they may be composed of water, proteins and polysaccharides. And extracellular matrix is highly dynamic structure that is constantly being remodeled. Okay, remodeled. And major components of the extracellular matrix are like collagen, elastin and fibrillin. Okay, the major components of the extracellular matrix are collagen, elastin, fibrillin, and special proteins are there, fibronectin and laminin, and few various proteoglycans. Okay, so my interest is to explain you collagen, elastin and fibrillin. Okay, these three are the collagen, elastin and fibrillin are the these are the components of extracellular matrix. And you know that what is extracellular matrix? These are the substances which has been surrounding the cells. So the composition of the extracellular matrix that will determine the functioning of the cell. Now coming to the collagen. Collagen is the most important connective tissue protein. Okay, and it constitutes 25% of the total proteins in your body. And collagen is formed in special tissues like fibroblast in the connective tissue and osteoblast in the bone. Okay, 25% of your proteins are of collagen only. It is formed by osteoblast in the bone or fibroblast in the connective tissue. And collagen is mainly present in the bone, teeth, tendons, cartilage and blood vessels. Okay, please note down, collagen is important short answer question for you. So you have to write all the points which I am mentioning now. So it's mainly present in bones, teeth, tendons, cartilage and blood vessels. And the composition of the collagen is here. It is the mature collagen. Already collagen is a protein. Obviously it is made up of amino acids. So nearly 1000 amino acids may be there. But every third amino acids in the structure of collagen is the glycine. Okay, every third amino acid in the structure of the collagen means 33% of the collagen is made up of glycine only. And along with that proline, hydroxyproline is there. And other amino acids like lysine, hydroxylysine, arginine and alanine. Okay, so glycine forms 33% of the collagen, proline and hydroxyproline, they constitute 21% of the collagen, and remaining amino acids may be lysine, hydroxylysine, arginine and alanine. And collagen is mainly synthesized in the fibroblasts. I told you, fibroblasts of the connective tissue or the osteoblasts of the bone from the precursor procollagen. Okay, collagen is synthesized from the precursor procollagen. After synthesis, it is secreted outside the cell. Once procollagen is synthesized in the osteoblasts of the bone or the fibroblasts of the connective tissue, they will be released into the cell The extracellular procollagen is then cleaved by the specific peptidases to form tropocollagen. Okay, originally collagen is not directly synthesized, first it is synthesized as procollagen. Once it is synthesized, it will be released into the cell. There procollagen undergoes released out of the cell in the extracellular space. The procollagen undergoes further modification by the enzyme peptidase to form tropocollagen, procollagen to tropocollagen. Okay, and that tropocollagen, if you see the structure of the tropocollagen is it's a triple helical structure. Okay, three polypeptide chains, they may be rounded each other, and each polypeptide chain is a rounded each other in a right-handed helix, and these tropocollagens are held by the hydrogen bonds and covalent linkages. Okay, imagine that 3 threads are there, they will be wounding each other, and how they will be attached to each other means through the hydrogen bonds. So there is a covalent crosslinks are formed by these structure is stabilized by lysyl oxidase. Okay, so like that how the first procollagen to tropocollagen, and you know the structure of the tropocollagen is a triple helical structure. Now here each of the 3 polypeptide chains tropocollagen is synthesized as pre-procollagen, and this pre-procollagen is undergoes translational modification, post-translation modification, that will be converted to pre-procollagen is converted to procollagen by the hydroxylation enzyme, and this is catalyzed by the prolyl hydroxylase and lysyl hydroxylase. Okay, now procollagen is then converted to mature collagen, and remember that for all the maturation of the collagen from the precursor, vitamin C is required. Okay, vitamin C is required for the maturation of the collagen from the tropocollagen. Tropocollagen is in turn derived from the pre-procollagen to procollagen. Okay, that's what, even in the deficiency of the vitamin C also there is poor hydroxylation occurs, which causes scurvy. Okay, and the next one is the functions of the collagen is this is the structure of the collagen. See here, this is amino acid sequence. Every third amino acid will be there the glycine. Okay, and this undergoes modification to form the collagen structure, triple helical structure, and they are further modified to collagen fibers. And here this mainly function of the collagen is 4 functions, just note down. Collagen is a connective tissue protein, extracellular protein, and collagen is made up of amino acids, and you know the composition, every 33% is made up of glycine only. Along with that, hydroxyproline, hydroxylysine, proline are there in the structure of the collagen, and collagen is it is synthesized in the osteoblasts of the bone and fibroblasts of the connective tissue. So there it is synthesized as pre-procollagen, which undergoes modification to form pre-procollagen to procollagen, procollagen to tropocollagen, tropocollagen is finally matured to collagen. For this maturation, vitamin C is required. And what are the functions of the collagen is mainly collagen which gives support to the organs and wound healing process. Okay, it mainly supports to the organs, and it has a role in the wound healing process, and in the thrombus formation in blood vessels collagen is exposed, the platelets are adhere and forms the thrombus formation. And anchorage effects, collagen provides the anchorage to the cells which help to in the proliferation and differentiation of the cell. Okay, these are the 4: support of the cells, wound healing, and it provides anchorage of the cells, and it has a role in the thrombus formation. Okay, and if there is any abnormality in the collagen structure and function, that may be result in the syndromes, that is the Ehlers-Danlos syndrome, osteogenesis imperfecta, scurvy. If there is any abnormalities in the collagen synthesis, may be associated with the abnormalities like Ehlers-Danlos syndrome, osteogenesis imperfecta, and scurvy. These are the disorders. And the next one, coming to the elastin. Elastin is also extracellular protein. It provides elasticity to the tissues, and which are constantly undergo repeated stretching. Elastin means it gives stretching to the tissues, and elastin is also a extracellular protein. Elastin is tightly associated with the collagen fibers. Okay, this is tightly associated with the collagen fibers. And here connective tissue protein present in the elastic fibers, mainly elastin where it is present in the elastic fibers of the lungs, large arterial vessels like aorta, and major amino acids which are present in the like elastin are glycine, proline, and alanine. hydroxyproline is present, but hydroxylysine is absent. Okay, what about here collagen? Hydroxyproline, hydroxylysine both are present in the collagen, but in the elastin, amino acids, glycine, proline, alanine will be there, but hydroxylated proline is there, but lysine is absent, and it does not form triple helical structure. Okay, because collagen forms a triple helical structure, but elastin it does not form the triple helical structure, and it does not contain carbohydrates. And here the cross-linking are formed by the desmosines. Okay, and this main function of the elastin is which is responsible for extensibility and elasticity in the lungs, which help in the extensibility and elasticity, and this elastin it confers extensibility and recoiling of the lungs, blood vessels and ligaments. Okay, that is the function of the collagen. You just compare here, this is the comparison between the collagen and elastin. Collagen is found in the skin and the protective tissue, whereas elastin is found in the connective tissue of elastic structures. Collagen is the third most abundant protein in the body, but whereas elastin is less abundant. Collagen is the white colored one, whereas elastin is yellow in color. Collagen gives strength to the structures, but elastin provides elasticity. Collagen gives strength to the structures, but elastin which provides elasticity. And collagen is produced until the aging process begins, but elastin is produced mainly the in the fetal period only. Okay, these are the points collagen versus elastin. And then one more is there keratin. Keratin is mainly found in another connective tissue protein. Till now we have discussed about the collagen, elastin, and then coming to the keratin. All these are the short answer questions. Okay, keratin is found in the skin, hair, nails, and the fibers are alpha keratins. And the matrix is here, and the types of it is mainly found in your skin, hair, and nails. The type of the fibers are alpha keratins are present, and each fibril has 3 polypeptide chains. As a keratin is a protein, it is made up of polypeptide chains. How many polypeptide chains are there in the structure of keratin is 3 polypeptide chains are there, and each bundle has 10 to 12 fibrils are there. And here mostly the keratin disulfide bonds are present here, and the covalent bonds between the lysine and glutamate forms the amide bonds. So the keratin of the hair has much more number of disulfide bonds, which gives strength. Okay, so keratin which is there in your hair has more number of the disulfide bonds are there, so which gives strengthening to the hair. So that's what unrestricting these bonds by reduction, the solubility increases while tensile strength decreases. This is the property used in artificial weaving of the hair. Okay, so hair strength is given by this disulfide bonds, but in the artificial weaving, so by the reduction, the disulfide bonds are reduced, and thereby the tensile strength is decreases. Okay, please note down the functions of the keratin is in the epidermis. It has a role in the keratinization, and it is an integral part of the structural network. Okay, of the desmosomes, and it is mainly maintain the spatial relationship between the nucleus and cytoplasmic organelles. Nucleus and cytoplasmic there is a spatial relation is there that is maintained by the keratin. It also has a role in the signaling, cell signaling. That means the information from the nucleus to the cell surface. So how they will be signaling, how they will be transported from the nucleus to the surface of the cell membrane is through keratin. Okay, these are the main functions of the keratin. Okay, and then see here, these are the other minor connective tissue proteins, fibrillin, laminin and fibronectin. Okay, you just concentrate on the keratin, its functions, collagen and its function, keratins and its functions. Okay, then coming to the last one is the glycoproteins and proteoglycans. So glycoproteins means here the carbohydrate with the protein, and proteoglycan is also protein with the carbohydrate. But how do you differentiate glycoproteins and proteoglycans? Means here the protein content is more in glycoproteins. Okay, protein content is more than the carbohydrate, they are called glycoproteins. If carbohydrate content is more than protein, they comes under proteoglycans. You just try to understand here. Glycoproteins means carbohydrate with the protein. Proteoglycans means proteins with the carbohydrate. So both the composition is similar only. So how do you differentiate whether it is a glycoprotein, whether it is a proteoglycan? Means based on their concentration. Okay, if protein concentration is more, they will comes under glycoprotein. If carbohydrate concentration is more, they will come under proteoglycans. More carbohydrate concentration, proteoglycans. More protein concentration than the carbohydrate, they are called glycoproteins. Okay, and glycoproteins usually glycosaminoglycans, mucopolysaccharides. In the carbohydrate chemistry we might have discussed mucopolysaccharides, glycosaminoglycans. They are the examples of proteoglycans. And most of the proteins which are present in your body, including albumin, is a glycoprotein. Okay, and you just see this one, glycoprotein means protein plus some carbohydrate. Proteoglycans means carbohydrate plus some protein. So more carbohydrate is there in proteoglycan, more proteins are there in the glycoproteins. Now here these glycoproteins, carbohydrate chains may be negatively charged, but in proteoglycans these are also negatively charged. But the examples of the proteoglycans are glycosaminoglycans. The carbohydrates which are present in the proteoglycans are mucopolysaccharides, that is glycosaminoglycans. But in the glycoproteins, usually the oligosaccharides may be attached to the glycoprotein. That's what carbohydrate content is less. Okay, in glycoprotein, protein may be attached to oligosaccharides. Because of the oligosaccharides, carbohydrate content is less. But in proteoglycans, proteins may be attached to mucopolysaccharides, heteropolysaccharides. That's what carbohydrate content is more. Okay, usually proteoglycans are long chain linear. They are not branched, negatively charged. But glycoproteins are branched one, usually they may be neutral or negatively charged. Okay, this is the structure of the glycoprotein, and they are the branched one. But this is the structure of the proteoglycan, they are the linear one, usually they may carry negative charge. Okay, here the proteins have been attached to mucopolysaccharides, here the protein may be attached to oligosaccharides. These are neutral or negatively charged, but proteoglycans are strictly negatively charged. Okay, so you just copy this. This is the comparison between the proteoglycans and glycoproteins, and here the first one is the core protein is covalently to one or more glycosaminoglycans. This is the proteoglycan. The first one is the proteoglycan, and this is the glycoproteins. Okay, function of the proteoglycan is they may be combined with the collagen to form cartilage and modulation of the cellular development. Function of the glycoproteins is they help in the cell-to-cell recognition and cell signaling. Where proteoglycans are present, mostly proteoglycans are present in connective tissue and glycoproteins are present on the cell surface. Okay, proteoglycan means here the carbohydrate content is more, they are present in the connective tissue. The carbohydrates are glycosaminoglycans and they may combine with the collagen. Their carbohydrate content is 50 to 60%. Mainly they may be associated with the water. They act as the shock absorbent and lubricant. Okay, as they are present in the synovial fluid of the joints, these proteoglycans, they act as the shock absorbents and lubricants. And okay, examples of the proteoglycans are chondroitin sulfate, dermatan sulfate, heparin sulfate, keratin. All these are the examples of the proteoglycans. Then coming to these are the glycoproteins. Here the carbohydrate content is less, only 10 to 15%, and they may be neutral or negatively charged. Okay, and the types of the glycoproteins are collagens, mucin, transferrin, immunoglobulins, etc. All these are the examples of the glycoproteins. Okay, with this proteoglycans like glycoproteins and proteoglycans has been finished for you. So with this let's say the one or two topics which are left over here in the lipid protein metabolism also I have finished. Okay, so with this your biochemistry syllabus is finished. All the best for your pre-final exam. Read well, go through your previous previous question papers. First finish off all the previous question papers. Okay, take the question papers of the previous 10 years like supplementary and regular main exams. Okay, and finish off all this one, and whatever the syllabus topics are there in your syllabus, try to finish off all the topics. Okay, all the best for your exam. Thank you.